Uh, Welcome, Arpita, to Parur University. You gave us so honorable time and you ready to give the presentation. So first of all, I would like to present you your short CV and then you can start your webinar. Okay. So Arpita Dave, she's Dr. Arpita Dave. She's currently working as a postdoctoral fellow in Jules Stein Eye Institute, UCLA. Her research focus is to comprehend molecular mechanism underlying retinal pigment epithelial cells impairment in early and late, that is age-related thing, and retinal degenerative blinding diseases. For her postdoc research, she is utilizing the most advanced RNA sequencing, reduced representation, bisulfite sequencing technologies. She is also involved in volunteering programs like teaching school kids about the eye anatomy and journal club discussion with graduate students also. She completed her PhD in zoology from MS University in Baroda, that is in year 2019. Her research focused on therapeutic aspect of flavonoid and POFA in inflammation and Rett syndrome associated diseases, primarily glial cell models. Some of the interesting findings got published in Journal of Molecular Neuroscience or communicated in conferences at Chicago, SFN, Parag, that is FENS, Chandigarh IAN, Chennai SNCI, and so on. During her PhD, she also received international and national travel grants to receive trainings on electrophysiology, stem cell biology, and rodent animal models. Due to her inclination towards scientific writing, she also freelanced for DNA technology. A reference handbook published by ABCCLIO, an American publishing group. She did MSc in biochemistry and BSc in zoology from MS University itself in Baroda. She was having a teaching experience of 1 year in PM Patel Paramedical College in subject biochemistry in year 2011. Her as I think her introduction is very attractive towards the curriculum, as well as she is also interesting for the extracurricular activity. Like her hobbies are reading and astrophysics, piano and music, Bharatanatyam, traveling to have a perspective on cultural diversities. So I think this was a short description as we are shorting of time, we are running out of time. This is the short description about the Dr. Arpita Dave. I would like to ask you, Arpita, please continue for the session.
Thank you, Indrani. Thank you so much for this wonderful opportunity and the introduction. So, for today's talk, I have outlined my presentation into two parts. In the first part, I would be discussing on major neurological disorders, neuroscience, basically a little bit about the nervous system, history of neuroscience, and I would like everyone to appreciate that it's not a very recent science. It has been studied, and people had curiosity in the past years as well about this field. Little bit about the models studied in the neuroscience field and recent technologies in using the electrophysiology, microscopy, and gene expression analysis approaches. In part two, I would like to discuss more about my PhD research, and in that I mainly focused on epigenetic regulation. And little bit I would discuss about one of the epigenetic factors' role in mitochondrial dysfunction in one of the glial cells, and one of the flavonoids effect I found in this disease model. So there are different types of neurological disorders known till date. Mainly they are divided into genetic or neurodevelopmental neuro disorder and injury, infection mediated or age related neurodegenerative diseases. And if you see, you might be aware of some of these disorders, like epilepsy is considered to be the one of the genetic disorder. Then attention deficits, Huntington disease, and Tourette syndrome. In one of the movies you must have heard about. Age-related disorders like Alzheimer's, Parkinson's disease, multiple sclerosis, which is the myelination disorder, migraine, ischemic stroke, and some of the psychiatric disorders like depression, anxiety, schizophrenia, and so on. So this is just a very short list of those neurological disorders, but there are so many actually. And if you see the global burden of these neurological disorders, then you would find that a large population has this stroke as one of the neurological disorder, and Alzheimer's disease and meningitis, which is the inflammation in the meninges part of the brain, migraine. These are very prevalent, not only globally but also in the Indian population. In the northwest India of the country, they studied all these things in recent times. So so that is very highly prevalent. These neurological disorders are in India as well as most of the countries. And if you see, I mean, recent thing is COVID-19. So I just thought of introducing some of those neurological manifestations related to COVID-19. And you must have heard that people have this headache, seizures, taste and smell impairment, which all are some or other way connected to neurons or nervous system. And that is why it is very important to understand the mechanisms of how this nervous system works. And briefly, I would say this nervous system is divided into two, for the simplicity, they are divided into two parts, central nervous system and peripheral nervous system. Central nervous system has brain and spinal cord. Peripheral nervous system is all other neurons innervating other different parts of the body. And if you see at the cellular level, these tissues have two major kind of cells, neurons and glia. And in the central nervous system, basically this oligodendrocyte, ependymal cells, microglia, and astrocyte glial cells are present, where basically they are the supporting cells. PNS has Schwann cells and satellite cells, and different types of neurons are present in all this in both the part of the nervous system. So this is a brief introduction of the nervous system. And if you see the history of neuroscience, you would really appreciate that it is not like this talk is about the recent findings, but this study started way back in 19th century. And if you see, with in those times, there were these technologies were not much developed. Only the light microscopy, and you see here the nitrate staining. Those very humble methods, they tried to understand the neuroanatomy, and this is cerebral cortex part. They tried to section the cerebral cortex and found a very nice systematic view of this brain tissue. In the 19th century, late 19th century itself, Francis Gotch actually found that this mammalian brain was capable of producing electrical current also, and which not all the not just the brain, but the nervous system actually basically had this capacity to produce electric current, which not other system had. And this Dr. Eric Richard Kandel, you must have read his book, or you must be referring to it. He gave really good understanding about learning and memory, and he established the fact that all the neurons are connected by synaptic through the synapses, and those specific neural circuits play a role in learning, memory, and many other functions. So with that, I would like to introduce two different models of studied in neuroscience. So different scientists have used human model, monkey model, rodents, zebrafish, Drosophila. They have used C. elegans also in some of the studies because of the simplicity of the animals. C. elegans and those C. elegans and those invertebrate models have less number of cells and less number of neurons basically, and that makes it easier to understand. And this rodent model is predominantly used because of its because they are more related in evolutionary to humans, and they can be studied in vivo also. So in recent times, what researchers do is they genetically manipulate some of the target genes to make animal model and to learn about the disease. So this three model, like there are different models you can see here in the slide. To I mean, in a different way, they researchers manipulated those genes to understand the disease condition and the pathogenesis associated with those genetic diseases. So but then I would like to highlight just these three because they are principally used in understanding the neurological disorder, and one of them is inducible and conditional genetic variation, in which what they do is they actually put that target gene under the specific regulator, so they can basically on and off the transcription and can see the behavior or different parameters associated with that genes. The other part is knockout, so in that specific gene is completely removed from the nervous system. Other is transgenic, where exogenously they provide some DNA and higher expression of that gene. How that higher expression of that gene can have a role in pathogenesis of the disease that they study. So this is about the in vivo model. Next, you can say that it's very like of course the rodent models are available, but then it's not that's not all, because it's very important to know the relevance in the human condition, and of course we cannot use humans as in vivo model. So what researchers came up with was human brain cells, and what one of the very important part is organoid culture, where they exploited this human pluripotent stem cell property, and what they tried was they provided some of the growth factors and guided this human pluripotent stem cell to transform into specific tissue and a specific cell type, and then in those cells actually then they introduce into oil droplet, and in specific culture condition in bioreactor they allowed this cells to actually assemble and grow into specific tissue type, and this basically then ultimately what comes up is very much resembling to the embryonic human brain. So this technique more helped in the disorders associated with neurodevelopment. So developmental disorders they could study very well in this kind of organoid, I mean this kind of model. Other human, human cells they use is the somatic cells from patients and healthy individuals. In that iPSC technology, what they do is they transform this somatic cells into like fibroblasts or something. So they transform those cells into undifferentiated cells, and then they are um, uh, they are directed towards specific cell type, and in those specific cell types, then they study the disease condition by checking the different parameters like the gene expression or anything which could be associated with that disease condition. Another thing they use as a model is primary cell culture, which actually complements the other part of the study. And in that, what they do is they isolate the tissue and in optimum medium condition and medium, they allow cells to grow in confluency, and then they can do some of the experiments in those cells. So molecular level of the study is generally preferred in the cell culture system, primary cell culture system. Now, nervous system study is broadly divided into these three types of tools or technologies, and now I would be discussing each of them in detail. So, like in electrophysiology, so the new most technique was optogenetics. This received this was invented by I mean majorly focused on by Dr. Boyden and Desserol, and they received Nobel Prize for this discovery. So in this, what they did was that some of the neurons, so as I said, that neural circuits are associated. They found that neural circuits like play a very important role in behavior of these animals. So what they did was those specific neural circuit associating neurons, they targeted, they provided, they transferred them with the gene which responds to the light. So one of those gene which responds to the light is channel rhodopsin. So what they did was when they provide the light, only those neurons or neural circuits which has those genes, which has those protein expression of those particular light sensitive gene, they would be showing the neural impulse, and they will be activated, and these neural circuits associated functions or behavior would be expressed by this animal. So that is how they could connect the specific neural circuits to the specific behavior of the animal. So that was the importance of optogenetic technique. Another interesting finding was this Miniscope recording actually. So Miniscope is nothing but the miniatured fluorescence microscope. So in that, I would say earlier people actually used to, in the 19th, 20th century, people had came up with very good technologies to record or visualize brain of the mice. But then the limitation in those technologies was that since they want, if you want to check the, if you want to see, visualize those neurons and record them, then microscopy was very, I mean, if you want to use the microscopy, you have to anesthetize the animal. So if you anesthetize the animal, and if the animal is not freely moving, then in a way you're compromising the behavior, because in anesthetized condition, the neural circuits might not play the way it plays in unanesthetized or free, unrestrained environment. So what this technology did was it allowed animals to freely move without any constraint, and this mini microscope allowed the visualization of those neurons in superficial layer as well as the deeper layer of the brain. And they also actually came up with another kind of this mini microscope which had no wire, so that allowed animals to have other social behavior also, because this kind of wire would not allow socializing of the animals in studies like anxiety and social behavior, depression kind of study. So when they removed this wire, they could actually using this micro SD card and battery system they incorporated in this mini microscope, they could actually visualize different neural activity in animal models associated. Next, in microscopy, one of the very interesting finding I would like to discuss is clarity. So in this clarity method, there was a constraint of the visualization of the brain, and that constraint was that this tissue, brain tissue, was opaque, so that did not allow clear visualization of the neurons. So in this method, what Carl Lisser did was he removed some of the plasma membrane. He removed the plasma membrane lipids in a way that the tissue, in a way that none other molecules are affected, and that allowed clear visualization of the brain, and that actually helped to immunostain those neurons, and it allowed the high resolution of 3D imaging without damage to the sample. So that technique was the breakthrough in neuroscience. Other recent technology is 3D electron microscopy, in which these tissues are dissected, and this technology allows individual brain cells, means nanoscale level of resolution, and as well as the large volume of brain tissue. So it allows us actually the smaller scale visualization in a large amount of a large size of the tissue, and that gives a very perspective idea about the tissue and molecular level, like tissue level and molecular level of the system. So that was the advantage of 3D electron microscopy. Another interesting technology that came up recently was combination of super resolution fluorescence microscopy and electron microscopy. And I would like to show this video where, I mean, I would not show the whole video, but basically they used electron microscopy, and this escalerol is green fluorescent protein which goes to the peroxisome only. So they use the fluorescence technology and electron microscopy, and they realized later on that with this method, they realized that these endosomes, which are yellow in color, they bind to peroxisomes, and so not all the peroxisomes are bound to endosomes. Some are bound to mitochondria also. So those variations they could actually visualize using this very innovative technology. And of course, you did electrophysiology to know the physiology of the cell. You did microscopy study to understand the anatomy of the cell. But another part is very important, and that is molecular level. So gene expression analysis of different ways is very important. Everyone must be aware of central dogma, and so in neuroscience as well, although in different diseases, the genome, transcriptome, and proteome studies are being done. I mean, there's different levels are being studied, expression is being studied, and I would like to introduce you to one of this genome study, which is genotype tissue expression and genome-wide association studies. In that, what they did was they in different donors in the postmortem tissues from different donors in different populations and the controls and in disease population, they isolated this different tissues, and what they did was they did genotype sequencing, genome sequencing. Also they did was RNA sequencing to understand the gene expression, and then they actually did the what do you call? They try to find which allele or genotype is responsible for the gene expression. So in that, I mean, in first example, you can see that this brain RNA is more in donor 1, and if you see that that is associated with this allelic condition. So from that, they got to know which genotype or allelic polymorphism is responsible for specific gene expression associated diseases. At transcriptome level, there are multiple technologies available now, like spatial transcriptomics I'm introducing here, but there is this single transcriptomics also, and cheap sequencing, and there are multiple technologies, but due to time constraint, I would not be able to discuss everything. And so briefly, I would like to just talk about the spatial transcriptomics, and that is like in spatial means location, space. So here what they do is they cut out the tissue, and that tissue slice they put on the slide, and in that slide itself they do cDNA synthesis and gene expression studies, and then they collect the data, and with the computational technology they establish in which part of the tissue which gene is expressed more or less. So that is how they can actually know that which part of the brain is affected, and how gene expression plays a role in specific condition or pathogenesis. Another thing is single cell proteomics, and also in the proteomics, which is the study of proteins expression, there are techniques, not only single cell, but spatial proteomics techniques is also available. So in very layman terms, I would say that so earlier there were the proteomics techniques, which was like smoothie, where all the cells are showing its effect, and then you see the cumulative proteome data. But single cell actually allows to cherry pick specific cell, and then in that specific cell you can know which protein is affected or not. So that is it. Briefly, this each cell is collected in well using this using the fats, and those collected those wells are then processed for protein sample for multiple for this mass spectroscopy. Each cell has actually its identity because of barcodes on that on the all the proteins of those specific cell, and that allows this single cell proteomic study possible. So that was the first part of my presentation, and I would like to say that as we see here, this puzzle with different technology and tools, people are trying to understand or solve this puzzle at nanoscale level, and if there is any other smaller level also of that, then that too. So that was the first part. Now, next I would like to talk a little bit about my PhD work. So it was so the PhD topic was role of flavonoid and polyunsaturated fatty acid in glial cell functions and neuroinflammation. And I'm going to present here a very small part of my PhD research. So I've not included everything about my PhD work. So a very small part was had this epigenetic regulation associated. So some of you might be knowing what epigenetic regulation is, but whoever doesn't know it, I would like to briefly, in a nutshell, I would like to tell them what it is. So in the chromosome, this gene expression or transcription is regulated by some of the DNA modification or histone modification. So in DNA modification, this DNA methylation is very important. And what DNA methylation is, you must be aware of the promoter region. I mean, each gene has its promoter region, and that promoter region basically allows the level of expression. So whether that gene should be expressed constitutively or episodically, that is decided by the promoter region, and this promoter regions have CPG islands. So like CPG islands are basically continuous CG region, and there this that CPG region, this DNA, some of the methyl transfer enzymes would modify those cytosines to form methyl CPG region, and that would actually help in transcription of the in the transcription to form mRNA and so on. For this DNA methylation related transcription, some of the transcription factors or epigenetic factors play a very important role in regulating this whole function. And one of those transcription factor is methyl CpG binding protein 2, in short MECP2, which is associated with Rett syndrome. And Rett syndrome is a neurodevelopmental disorder, which is X-linked disorder, and it causes mental retardation and some of the sensory motor function deficits and things like that in females, since it's X-linked disorder. In my studies, I studied that gene, this MECP2's role in glial cells, and I tried to see if this MECP2 can affect the mitochondrial function. Especially, I checked oxidative phosphorylation in mitochondria, because mitochondria has definitely not just this role in ATP synthesis, but many other things. But my focus was just oxidative phosphorylation in mitochondrial function. So I tried to check in glial cells this role of this MECP2 in oxidative phosphorylation. The other part of the study was to see MECP2's role in calcium homeostasis, and for that I did cytosolic calcium measurement. And the third part of the study was to check whether this one of the flavonoid or natural compound quercetin, which is abundant in onion. So that compound has any effect on this mitochondrial function mediated by MECP2 and calcium homeostasis, which is mediated by MECP2. So Um, for this oxidative phosphorylation study, I checked enzyme activity in negative control in MSCP2 knockdown cells, and I used 2 different doses of quercetin. In those cells, in those conditions, I found that this enzyme activities of complex 2, complex 3, and complex 4 was increased in dose-dependent manner in case of quercetin-treated um, groups. So next part of the study was related to the mitochondrial membrane potential, and everyone might not be aware of mitochondrial membrane potential. So a little bit about that. So mitochondria actually, because of this electron transport chain or oxidative phosphorylation chain, has removes hydrogen ions from the matrix, and that makes the inner side more negative. So when the inner side is more negative, that is the hyperpolarized state, and in that condition, the fluorescent dye which I used in my study would express more, like would fluoresce more. Uh, in depolarized state, which means that inner side is less negative, which means that less number of hydrogen ions are going out of this matrix. So in that condition, the less dye, because of its nature, chemical nature, would accumulate in the mitochondria. So in a way, when less fluorescence inside the mitochondria, oxidative phosphorylation is less, and if it is more fluorescent inside the mitochondria, then the activity is higher of this electron transport chain. So as I said earlier about this enzyme activities, in knockdown condition, the activities were reduced. Similar observation I could found in this mitochondrial membrane potential studies, that in the MCP2 knockdown group, this membrane potential was reduced, which means like this it was in the depolarized state, and interestingly, this quercetin treatment some way this mitochondrial membrane potential, as we see in also in the enzyme activities, that it was increased compared to the knockout condition. In calcium studies also, I could see that this quercetin had beneficial role, and so that's how I would suggest. I mean, this part of the study, which was published into 2019, suggested that quercetin can be actually focused on for conditions like Rett syndrome, and I mean, this was just the in vitro study, but in vivo study in future would help us to know if quercetin can be used as a therapeutic participant or not. So that was about the PhD study, and yeah, I almost completed my presentation in time. I would like to acknowledge Dr. Prakash Pillai, my PhD mentor at MS University of Baroda, Professor Vibhakar Kotak, who actually I know him because he presented in MS University once upon a time, and I contacted him to learn more about the computational neuroscience, because at that time I was more interested in computational neuroscience. And then after that, I discussed many things about neuroscience with him. So that's how I got really good opportunity to learn many things about neuroscience. Dr. Roxana Radu, who is my current mentor in Jules Stein Eye Institute, to the viewers, I would like to introduce to the training opportunities also. If anyone is interested in neuroscience, and I myself have attended some of this training programs. They actually give grants to learn those technologies, new technologies in neuroscience. And I have attended this IBRO organized training program, FENS organized training program, and also the SFN thing, and this IAN, and this is the national, Indian groups, and I have attended those workshops as well. And this is like just to of course we learn scientific things in workshops, but with that we get the opportunity to meet different people which we do not otherwise get. So in a FENS organized workshop where I learned about the stem cell biology, I get a chance to meet people from different countries. So this is Prague, where I went to learn about the stem cell biology. I went to Chicago for the SFN, Society for Neuroscience conference, where I learned more about the rat syndrome, and I presented my PhD work there. In Tehran in 2017, I learned about the electrophysiology, so I did the mini project, and that was also part of this training opportunity. And this is my postdoc mentor. This is one of the volunteering opportunities I got in Julestein, where I worked for vision awareness. This is the Julestein Institute, and this is LA. Anyway, and thank you. And I thought of playing a quiz kind of a thing to know if people can answer these questions, but since I cannot see anyone on this Google Meet, I would just keep this part. Thank you. And how do I get to know that I'm done?
Thank you so much, Arpita.
Okay. So can people ask questions to me here, or how's it?
Actually, they cannot directly ask any question, but I am asking. We can wait for 5 minutes. I am giving them 5 minutes time. So within that 5 minutes, if someone is having any question, they will text me. So the same question I will be asking you. So we can wait for 5 minutes.
Yeah, even I would like to say that if someone wants to apply to any of these training opportunities, or even if someone wants to do masters outside in some other country or something, and they need any guidance from my end for writing research statement or SOP, I would love it. And here is my email address, so anyone can contact me, and I would definitely respond. Yeah.
Yeah, so I think for BSc students, it will be really beneficial. As already Dr. Arpita has mentioned about her email ID also. So if anyone have any query or you want to ask any type of questions, please type in my chat box in WhatsApp so that I can ask question to her, and she will be able, and I think she will be very happy to answer your questions. So many students over here who is in the last semester of graduation, they are eager to go outside, right? They are eager to work outside India, and they are trying for their masters in out of India. So I think this you will be getting a good opportunity, and she is there to guide you even.
Yeah, happily.
So guys, if you have any question, please ask the questions. We can wait for 2 minutes so that other students or any faculty members, anyone who is who want or you who have any query, they can just text me.
I actually can't see anyone, so obviously I don't know whether people followed by what I discussed or not, but I am assuming
Yeah,
that
yeah, actually,
somebody got
yeah, actually
something
we
and missed
can
out.
see. No, no, in Facebook also, there have lot of comments. You can see the students and faculty members are attending that.
Okay.
And it has been a live session, so everyone is there.
Mhm.
The thing is that on the platform, each and everyone cannot be there. The thing is that many students get interruption. They will cause some interruption. So it is difficult to manage for the host as well as for the guest. So that is the
Mhm.
reason we are only keeping webinar people, few webinar, host and the guest, the
Okay.
speaker.
Mhm.
Okay, one question has been there. What are the examples of epigenetics? What are the examples of epigenetics?
What do you mean by example? What are other modifications?
I think she wants that only. So sorry.
Okay. So I hope a person know what epigenetics is, and if you do not know, then epigenetics is basically um, um, some modification in not the genome, but like it doesn't have any permanent genomic modification, or it is not inherent thing, but some modification in a way which can have which has role in transcription or just the gene expression. So in histone modification, there are different types like acetylation, ubiquitination, sumonylation, and another modification is methylation as well. So these are this histone modification in which those molecules, acetyl group or ubiquitin group, they bind to histones, and that allows structure this chromosome to be in heterochromatin form or euchromatin form, and that in a way regulates the transcription of those genes. I did not discuss that part. Since my study was on the DNA methylation, so in promoter region, in the CPG island of the promoter region, methyl group binds with the help of methyl transferases, and there this transcription factors play a very important role. Yeah, I hope I answered the question. Hi. Oh, am I still there?
Another question. Yeah, you are there. Second question has been asked. I hope the answer has been there. You got the answer. She got the answer. So,
Okay.
second question asked, that is, can epigenetic changes be passed from generation to generation within a family or not?
Actually, that's very interesting thing, and in some of the recent findings, they have said that this DNA methylation footprints are there in the next generation. So, how I do not know, but yes, that footprints of epigenetic regulations are inherent somehow. Yes, so they can pass generation to generation as well. Yeah.
Okay. Next question. What are the factors that is going to affect the epigenetics?
Factors, yeah, there are so many factors: lifestyle, smoking, environment. There are so many. Stress is one of the factors. Aging is one of the factors. So yeah, there are so many factors.
Okay, and one student, I think she had asked, what are the three major epigenetic mechanisms? So I think she had already given her answer in the
Okay.
conversation only, that DNA methylation, histone modification, and all that are there. But you can give
Mhm.
answer. What are the major epigenetic mechanisms?
What I know is like more is the histone methylation, histone acetylation, and DNA methylation. These three are, I would say, more major. And there are other also, I say, ubiquitination, sumonylation, but they are not much studied yet.
Okay, another question. Is gene slicing or its activation is related to epigenetics?
Yeah, oh yes, of course. Yes, so some of, thank you for reminding that to me. Yes, splicing is also part of the epigenetics. Yes. Yeah, I have not put everything here because I just wanted audience to direct to my research. So yeah, splicing is also the part of epigenetic regulation. Thank you.
So sorry. I think this many questions are there. That's it.
Okay.
Still, if any audience, if they are having any questions, they can text me or you can directly put your questions to her mail ID and she will be giving you answer. Or if someone is hesitating, they can directly ping me so that I will forward direct question to her and she will be happy to answer your questions. So thank you, Arpita. Thank you so much on behalf of Parul Institute of Applied Sciences, Biotechnology Department. You gave your valuable time, and I hope that all of us got some of good information from you, and it will be beneficial for the students as well as for the faculties of Applied Sciences and to all the viewers who had viewed this session.
Thank you.